we're going to start using the information that we've talked at the beginning, and but I want to take you on a different type of journey. I think this issue of oxygen is an issue where neonatologists have a love and hate relationship with oxygen. Yes, we love it because it helps our babies, but at the same time we hate it because we've seen the negative effects. And so what I've done a couple of years ago is started to inquire more about oxygen and to try to understand what the rationale is for what we do today with oxygen. But to do that, I have to go back in time to then come to where we are. And so I want to take you a little bit through that journey because I think that's something that all neonatologists should look at. After this type of presentations, I've had a lot of colleagues that have started to do research and to look into this on their own, and it becomes like an ongoing path because the research that's going on at the present time around oxygen and reactive oxygen species is tremendous, and how it affects genes and gene switching, and so it's a never-ending process of learning. So I'm going to go through that with you today. So I think the battery might be out. It doesn't. Oh, it's not. You need me to put this back on this, right? Okay, I'll change it. Do you have okay. Oh, we took that off on purpose. Okay, so when I told you about the first randomized controlled trial in neonatology, it was looking at retrolental fibroplasia, ROP. And this was the trial that was done published in 1954. And basically what they did during a one-year period, it's a cooperative study where they took babies less than 1,500 grams, survival at 48 hours. And they looked at two groups of babies, ones that were randomized to the conventional group, which was 50% oxygen, and the other control group that were weaned very quickly to room air. And then they looked at the outcome of ROP, and they were able to demonstrate that the ones that were exposed to the 50% oxygen had a significantly higher incidence of ROP compared to the babies that were not exposed to that 50% that were curtailed and weaned very quickly. And so and these babies were all randomized at 48 hours of birth, and then moved, they were observed during a 28-day period. And so this was very important in understanding that oxygen, yes, very good, but you can have negative effects with respect to oxygen. And of course, this was supported then by mice models of ROP where they were exposed to different levels of oxygen, hyperoxia versus hypoxia versus normoxia. And then they were able to look at the retinas and what happened with the animals that were exposed to hyperoxia. You can see that there's thinning of the vessels, less vascularization. And so hypoxia with the hyperoxia, what you were producing there is a sense of hypoxia to the tissues. And so that hypoxia then led to increase in growth factors and to proliferation of vessels, neovascularization, and what we understand today of ROP. But you can see the differences between normoxia and what you do when you expose the animals to hyperoxia. And then this paper came out from the Karolinska Institute in 1959, where they took mice and they exposed them to 80-something% oxygen for prolonged periods of time. And what they started to notice that these mice started to die after 48 hours, 72 hours of being exposed to 85% oxygen. Nothing else, just exposure to that percentage of oxygen. These animals died. They did the autopsies of the lungs of these animals, and they had severe pulmonary edema, pulmonary hemorrhage, and there was inflammation in the lungs of those animals. In 1963, I believe this was the case where the 32-weeker of John F. Kennedy was born. And it was very interesting. That baby was born premature at that time, what we would closely include as those late preterm infants, maybe if we include 32 to 36 weekers. It was a baby that had respiratory distress syndrome. And the baby was born at a military base and then was transferred to Boston and went first to Harvard, to the Boston Children's, and they didn't really have a good way to treat the baby. The baby was getting worse. And so they transferred him to the School of Public Health. And in the School of Public Health, they put the baby in a hyperoxic chamber and exposed him to high pressures of high oxygen concentration, and then the child died shortly after that. And so it was a time when we didn't understand the pathophysiology of respiratory distress. We didn't have, the only thing that we had was cannula, oxygen. And so it was thought that this might give this baby an opportunity. Today, in retrospect, it's sort of the repetition of the experiment with the mice exposing to high concentrations of oxygen. You can produce more lung damage, and you can produce pulmonary edema, and maybe it worsened the outcome of that baby. And so little by little, we began to understand a little bit about the pathology, and this is Northway and Roseanne pathologists looking at what was happening with babies that in the 60s were the first time that we started to put babies on mechanical ventilation, and we would give them a lot of high concentrations of oxygens. The babies were surviving, but there were a group of babies that did not survive and died, and they were able to look at that pathology and describe all those findings. As you know, what we now consider in that time bronchopulmonary dysplasia. We're using that concept still, chronic lung disease. And then you've seen all the discussions that we're having at the present time about how much oxygen should we be exposing premature babies to. And so, as I mentioned, 5 years ago, all of us were putting babies when they were born, resuscitating them on 100%. And the NRP training included 100% oxygen for all these babies. How did we start to do these trials? Well, it was very interesting because they came from developing countries. And so it came from India, because in India they started to do NRP training. But because they couldn't have oxygen in those sites where they were training the nurses and the midwives to do NRP training, they were resuscitating babies on room air. And so I was at one of the Scientific Pediatric Society meetings at one time, and when they presented those studies, and we had just come from Guatemala from training midwives there in NRP, also on room air. And I remember one of the doctors that was there that was so upset because from India they were presenting this study where they were not exposing the babies to oxygen. And he said, this is unethical, for them not to be giving babies oxygen. And he just stood and he left. And I started to think about what he said, and I said, well, oxygen, 21% oxygen is oxygen. We're not just not giving oxygen to babies, they're getting 21%. Maybe that's insufficient, but maybe it isn't. And so that question, because they were showing that they had increased their survival with that new protocol that they were using there, I think that question was raised, and then somebody went and started all that research that's being done today with respect to that. And then you had the support trial where they had two groups that were randomized within that CPAP trial to a low target saturations versus high. And you can see that, yes, there were differences, but there were not humongous differences. If you were in Colombia, where I come from, in Bogota, where we're 2,600 meters above sea level, having SATs 85 to 89, 90% in a preterm baby would not be anything that would be surprising for us, because that's how the majority of the babies at that altitude, that's their oxygen. If you measure term infants only, and you measure their saturation, and that study has been done at that altitude, average 93. And so for us, it wasn't the same, but they tried to simulate that in that second group at sea level, and what they found was that there was increased mortality, decrease in ROP. again, the concept of a double-edged sword. You're going to cut and bleed on both sides. Where is it that you want to bleed more? And so mortality, you don't want to tolerate that type of bleeding. Maybe you have to tolerate more ROP, but there's no way to get away from these diseases. But you can see that just a shift in the way you target your sats in the nursery can affect your outcome. Individually, as a NICU, we would never be able to answer that question. We would always think that we're doing the right thing. So you need, of course, these large multi-center trials to be able to come up with that. And so what I'd like to go back in time and look at oxygen and what oxygen means to us in the process of evolution, and to try to then use that information so that we can start rationalizing our use of oxygen. Some studies have been, that are now looking at, for example, the long-term effects of exposure to oxygen, high concentrations of oxygen in newborn infants, and some are finding correlations, associations, still not cause-effect, but correlations and associations between children having leukemia at 5 years of age and then being exposed to 100% when they were premature infants. So this has become something of a concern. And so what I'm going to show you will try to help us understand if there is some truth to what they might be proposing in these studies. And so we have to begin here, solar energy, and how we have that solar energy, the fusion of 2 neutrons, 2 protons, produce helium and light, and how that affects our planet and how important this process is. But here I'd like to make emphasis on 2 things, and is the presence of the ozone layer that we're so concerned about at the present time, and how that ozone layer is blocking these ultraviolet lights that really are the ones that produce damage and can produce cancer. We all know that. As this ozone layer starts to decrease or thin, and then we start having, of course, increase in skin cancer and other problems. And so what I want to take this information and then put it into the context of what happened 3.5 billion years ago. And so if we go 3.5 billion years ago to the Archean period, and here's a depiction of what our planet used to look like at that time. If we took a sample of the air and the water, we would find out that in the air there was no oxygen, and that the only thing that was there was methane and CO2. And if you look at the water and you took a sample from the water, you would find that there were live bacteria at that time, what we consider bacteria that we call prokaryotes, and they were proliferating in an environment that was at total absence of oxygen. And what did they use for their metabolism and to survive? Well, they used methane to be able to survive. That's what they incorporated in their metabolism to be able to do their metabolic functions. And this is what you would find a basic prokaryote is, and you can see the capsule, the cell wall, a cell membrane, a cytoplasm, and with these small organelles, ribosomes. Many of those ribosomes were in the inner layer of that membrane, and you had this RNA, DNA structure here that did not have an envelope around it. And what we learned after that is that many bacteria must have died because at that time, of course, no oxygen, no ozone layer. So ultraviolet radiation was hitting the surface of the earth and of that water. But these prokaryotes were surviving that exposure. Many of them must have died, millions of them, but there were some that were surviving. And then some of them learned to use that ultraviolet light to be able to do photosynthesis, take CO2 and water, CO2 and vapor water and ultraviolet light, and produce carbohydrates for their nutrition. And as a byproduct of this, produce oxygen. And so this is what is considered the cyanobacteria that was so successful proliferating in the planet and mostly in water. And this just summarizes this caricature, just showing light and oxygen, and this structure that these prokaryotes have developed, which is it's the basic structure of what we now see in plants, which was a chloroplast. And you can see they make their carbohydrates for their nutrition, so they don't need a gastrointestinal tract. And their product of elimination is oxygen. So what is believed now by some of the researchers is that as this cyanobacteria started to proliferate, the concentrations in the microenvironments in the oceans started to increase also. But oxygen in the presence of ultraviolet light will create reactive oxygen species, and we know we reproduce that all the time. So this oxygen became a toxic, and because of the formation of reactive oxygen species, many of those prokaryotes did not survive because of the presence of oxygen. And so there was a challenge in the environment to these new organisms that had been living without oxygen. But the ones that developed the capacity to use ultraviolet light and were able to develop those chloroplasts were able to survive. And these little structures that you see here are thylakoids. When you look at them, they're very similar to what you see in mitochondria. And they are basically transport mechanisms where basically synthesize ATP. They found a way to use their energy to conserve energy through the formation of ATP. So here again we go to our anaerobic bacteria, and you see oxygen coming into their cells, to their metabolism. Then you have these products, toxic products that are produced, superoxide dismutase, hydrogen peroxide, hydroxy radical. And then if they didn't have detoxifying pathways for these oxygen radicals, then they would die. Basically, death would be the outcome. And then what we see from an evolutionary process was that at one point in time, there were prokaryotes, ones that started to go deeper into the ocean, wanted to stay anaerobes, and we still find them there. And I'll give you some examples, because now we're reproducing the environment that we had on Earth in research 3.5 billion years ago. It's been done with all the research that's been done for Mars. And so what we're going to talk about is how that research correlates to what we're seeing here. But they somehow started to develop through evolution and mutations. They developed the proteins to make enzymes that would facilitate coupling oxygen to glycolysis and then formation of ATP without having chloroplasts, these type of prokaryotes. And in that process, they were able to create detoxifying enzymes like superoxide dismutase, catalase, peroxidase, and then neutralize these and then produce these byproducts. And at the same time, because they needed to have this going on, not stopping, that's when we developed the capacity to couple that to the ongoing formation of ATP to conserve energy. And so the investigators now call this prokaryote that was able to do this and not die when exposed to higher concentrations of oxygen, what we'll call here the alpha protobacteria. And in the theory today is, and we'll go through how we came to this, is that it was first this alpha protobacteria that what we understand is a mitochondria and that we have in all our cells, that at one point was an independent prokaryote that developed that capacity to use oxygen appropriately and create these detoxifying systems. And so going quickly to oxygen radicals, so you see a molecule of oxygen and with their 6 electrons, 2 in the center for it, and they have covalent bonds to make them very stable. But ultraviolet light makes them very, very unstable. And so at the beginning when they were forming these cyanobacteria, increasing slowly the concentration of oxygen in the oceans and then in the atmosphere, we did not have an ozone layer. Those ultraviolet lights, and we now know that's something also called desiccation, which means those prokaryotes were exposed to moments where the sea level went down, and desiccation would produce increase in exposure to ultraviolet lights and would produce more reactive oxygen species. And of course, many of those would not survive. So they would split these into 2 different atoms, and then because they were so unstable when they were not together, they would lose an electron and create a free radical. And so here's the power of oxygen, because we think that oxygen is a toxic to our patients, but we understand now that it's more the creation of oxygen radicals that are creating the toxicity. And so anything that stimulates exposure, if you for example, start to expose children to ultraviolet light, newborn infants, premature infants, and you start giving them supplemental oxygen, you're going to exhaust all their capacity to tolerate oxidative stress. And just with exposure to that light, you will produce more lung damage in those babies, something that we didn't understand in the past. And so we also now understand the concept of switched genes. By putting a baby on phototherapy, there are genes that are switched on and genes that are switched down. And some of these genes in experimental animal models, they've been shown that you can give an excessive dose of sugar to an animal, and then you expose them to a phototherapy type of light, and then you see that those babies become hyperglycemic, and they become resistant to insulin. When you shut off that light, then you give them the insulin and their glucose goes down. So a lot of things that we're learning and that we didn't know about, of how you can turn genes on and off, and how that can affect oxidative stress. And so these are the ones that we're going to be talking about. We're going to focus on them. And of course, we're especially interested also in that hydroxy radical, because in the presence of iron and through phenton kinetics, you form this hydroxy radical, and it's the most known powerful reactive oxygen species that can affect cell membranes, especially lipid membranes. So now we go to the Proterozoic period, 2.5 billion to 543 million years ago. And very interestingly, this is a point of discussion, and very, very interesting, of how prokaryotes then go to become eukaryotes, multicellular organisms. And so by that time, that's what they start to see, that around this period of time, for the first fossils of eukaryotes occurred. And so how did that transition occur? And so this is a stromatolite, and in these structures, which we still have today, that's where they're finding these new fossils and trying to study them. And what basically I'm showing you here is samples of 2 eukaryotes, and you can see their organelles that now they have that envelope structure of the nucleus on both sides. They have this endoplasmic reticulum, which we will talk about, because that endoplasmic reticulum is what gives origin to the nuclear envelope. And so there are now theories that are supporting the fact that they have shown that oxidative stress and desiccation can produce damage to the lipid membranes, and when they produce damage, to protect the cells themselves, they invaginate parts of the membrane, mostly of their inner cell membrane, and then detachments or vesicles that are formed from that, they pinch off, and there's a strong support that that's what created that endoplasmic reticulum. And they have proof of that because they've looked at the ribosomes that are in bacteria in their inner membrane, and the whole system of organization is very similar to what you find in the endoplasmic reticulum. You see the chloroplast here, and you see that there's mitochondria, and it's believed that those mitochondria are there to protect first from oxidative stress, and then second to facilitate all that process of forming energy, ATP, and moving forth glycolysis and metabolism. These eukaryotes have exactly the same structures but don't have chloroplasts. And so by necessity, they have to take nutrition from the environment to be able to survive. But you can see here what they show is that their endoplasmic reticulum is much, much, much more developed than what you see in these other eukaryotes that have chloroplasts. Their problem is resolved. They produce their own energy. They don't need a digestive system. And they don't need to breathe. They breathe through, all their anatomy is made to breathe. They have pores where they can do that. But for this eukaryote, they need to develop a respiratory system. And of course, you can see that they have much more mitochondria. And of course, part of that relationship between the nucleus and that endoplasmic reticulum. So here we put together photosynthesis and respiration, and you can see CO2, water, sugars, and plants and algae from the cyanobacteria, then we have the development of eukaryotes that are algae, and blue and green algae. And their source of energy, of course, they produce their own sugars. Here it's the opposite. For eukaryotes that don't have chloroplasts, you have to take sugars from the environment, and then with oxygen, which you need it, then you have water, CO2. And that's, of course, most living organisms. So the first hypothesis that was related first was that at one point we had prokaryotes without chloroplasts, and then they evolved and learned how to use ultraviolet light to their advantage and generated new proteins that facilitated the development of these chloroplasts and how they became independent with that with respect to their nutrition. And these were the prokaryotes without mitochondria and prokaryotes with mitochondria. That's part of the other evolutionary process, that these prokaryotes didn't develop chloroplasts, but they had to develop a system to be able to survive in higher oxygen concentrations. And that in that process, something happened that then permitted their evolutions towards eukaryotes. And so all of you have looked at this, the origin of species and how the environment affects survival, and the fittest are the ones that survive. Well, all that now is being used in microbiology and molecular biology to try to explain part of what we're talking about, and how interesting and how you can relate to that information. In the 1960s, this microbiologist here in the United States, Lynn Margulis, came up with a theory that initially was rejected by all the scientists. And they didn't pay attention to that theory. And 40 years later, all biogenetics is now reaffirming her theory. And basically, her paper was published a long time ago. It's called The Conscious Cell by Lynn Margulis. And her theory was that there was a bacteria, a primitive bacteria that was called an archaebacteria, that they call there a thermoplast. And it entered into what they call a symbiotic relationship with an eubacteria, which is the type of bacteria that we all know today, like E. coli. And we have, at the present time, we have archaebacteria that we are taking from volcanic vents that can tolerate large, extremely large temperatures, and that have the same characteristics of that archaebacteria that we're thinking of. And they are different in their composition and what they offer, but they believe that it was a symbiotic relationship between these bacteria that formed the first protist. And here, this graph shows it. An eubacteria, archaebacteria, they consort and they create this chimeric stage, and you have an archaeprotist, and then you have a protozoan. And you see that initially they don't have mitochondria, and then they acquire mitochondria. The process here is that this was also an independent bacteria that then was engulfed by this archaeprotist, and then incorporated it and did not destroy it because it became a mutually beneficial relationship. One produced the energy and protected them from oxidative stress, and the other part of the receptor cell also gave them a safe environment and gave nutrients. And this is what it must have looked like, the first eukaryote. And this just summarizes, they think that the process was done through endocytosis of a bacterium that had developed the capacity to use oxygen appropriately and protect themselves from oxidative stress. The host cell, and you see that that inner membrane is then what engulfs the outer structure of what now we understand is a mitochondria. And so that was the theory that received so much critique at the beginning. But of course, there are things that support that there is reality in what they were saying. And one of the things is that when they look at the DNA of mitochondria, it's very different, we all know, from the mitochondria of our cells. So these are circular DNA, and that circular DNA is very typical of all the bacteria that we know. That's one of the differences. And the way they reproduce by binary fission is it's the same as bacteria do it, while we have meiosis. And meiosis is and mitosis and all that process, what we're learning now in the research that's being done, they believe that that sex occurred precisely at that time where there was the engulfment of one bacteria by another for mutual advantages, and then how they shared that DNA, that process had to occur simultaneously for meiosis to have occurred and for fusion of gametes to have occurred. And so all the biomolecular research are now beginning to support that theory. This is a Russian paper that was published in 1924, and it was never translated to English, so nobody really had access to this book. And so Dr. Margulis, after many years, got a hold of this book. Her theory was not a new theory. This was already posed by somebody in 1924. And it was this gentleman, researcher from Russia, Kosovo Polinarsky. He was the first one to consider that endosymbiotic relationship. And how did he do it? By observation. He was able to look at how in the steppe in Siberia, what was the nourishment of all the mammals that were in that area. And he went back and found that as a commonality, it was lichen. And then he started to look at the lichen, and he found out that the lichen was not a plant, that it was the result of a symbiotic relationship with algae and fungus. And so that began his theory, and he wrote all this book about different endosymbiotic relationships and how they were mutually beneficial. And so if you put Darwin together, and I don't think Darwin was able to read this book and put what Polniowski had done his research, I think it would have led to a more rapid evolution of what we were trying to understand at the same time. And these are just examples of how we find today this endosymbiotic relationship. This is a flatworm that it has incorporated into their organism algae, and they use it to be able to produce their own carbohydrates. And so they don't destroy the algae, they just keep it and protect it, because the algae through photosynthesis gives them their carbohydrates. And so it's a mutually beneficial experience for them. And so there are multiple examples of these. If you start looking at that research, you'll find that there are many, many, many examples of those relationships. So today in the research area of biomolecular research, it is considered that both the chloroplast and the mitochondria were previously prokaryotes that learned how to use ultraviolet light independently, and others that used to learn how to use oxygen when oxygen levels became toxic. So all these are mechanisms of adaptation, and that's where we find that of survival to the environment, stresses in the environment. But what we're now understanding more is what modulates those changes towards adaptation. You have a stressor, you die, or you adapt. And so what is it that's making the changes? And so today we're learning that it's the reactive oxygen species, because every time we talk about reactive oxygen species, we're talking about the bad effects of oxidative stress. And definitely for many, many of these cells and organisms that were alive, it destroyed them. But at the same time, reactive oxygen species, from research that's now being done, it is the point in all evolution of how evolution moves forward, because that's what produces denaturalization of RNA and of DNA, or it kills it, or it helps create new types of proteins that facilitate the process of adaptation. And so that's what we're going to be talking about. So you have genetic stability, which is a good thing, but too much genetic stability doesn't let you evolve. And then you become, if the environment changes and you have a lot of genetic stability, you get extinct. And we have examples of that in nature. And so along with genetic stability, you need also genetic mutability for evolution. So mutations not only produce cancer, yes, that's a bad thing, but when we look back and how evolution has moved forward, these mutations in many cases have been very important, because they generate the creation of new proteins that permit us to create the enzymes that protect us from oxidative stress. But again, they create other mechanisms that help us adapt to more stressful environments. And this is sort of a graph that talks about the environment, the normal and stressful, and what it does genetically. And There are very specific examples that are coming out. This is a paper from Evolutionary Biology of 2010, and how important it is in some cases to be able to have switch genes that turn on that will facilitate creating new proteins or proteins that protect you from oxidative stress. And sometimes when those do not switch on, then you have disease, you have cell death, or your what you call programmed cell apoptosis, death, apoptosis. When you look at it, it's all moved forward through reactive oxygen species. The death of these cells is through the presence of these reactive oxygen species. So they are both helpful, but they can also be harmful. And now we have this new concept of ribose switch genes, and we have not only on RNA, but also DNA that are changing the way we look at and understand genes and how different factors in the environment can affect them. This is where we're getting these new bacteria that are prokaryotes from these volcanic sinks. And so now there's a whole gamma of research that's been done in environments where you can manipulate it to take away oxygen and take these bacteria along with the bacteria that we traditionally know as ill bacteria like E. coli, and then they expose them to very harsh environments, and they're trying to look at how their cell membranes change to see if they can reproduce that evolutionary process. And of course, this is one of them, Methanococcus janishi, taken from the base of those. These tolerate up to 250 degrees centigrade temperature, and nothing happens to them. They survive and thrive at that temperature. And they've been able to map all their genes completely. And so that's now where the new terrain for research is. So the hypothesis, this is another from this paper of Biology Direct 2010, is that a local rise in oxygen levels due to cyanobacterial photosynthesis in ancient arcane microenvironments was highly toxic to the surrounding biota, and their support for that. This selective pressure drove the transformation of an archaeal bacterial lineage into the first eukaryotes. That's the hypothesis that they're trying to demonstrate. And so in this very interesting paper, I would recommend that everybody reads this paper. It's just a fascinating paper on how they go through all that different processes. But here, this part here, basically is their hypothesis of how an environment that's stressed, for example, with hydrogen peroxide affects the external membrane where you find the ribosomes, and then this is where it invaginates to protect itself and then pinches off and then maintains the same structure of the inner membrane of that cell. And then from then you have all the endoplasmatic reticulum develop as an organelle. And here it looks at how two prokaryotes they basically, like bacteria, they make by conjugation, by fusion. And so their hypothesis is that one of these bacteria that develop the capacity to take oxygen and use it appropriately to produce antioxidants and to couple ATP, and form ATP, were basically put into a sandwich and then incorporated during the process of conjugation of these two prokaryotes. And then you have the development of all these mitochondria that then reproduce by binary fission. So the theories today is that oxygen is a driver of evolution, and that more specifically the reactive oxygen species were driving all the process of evolution to the point where we are. And so if we make the comparison, we have 10 times more bacteria in our gut than all the cells in our body. And you add to that concept that if all this is true, these hypotheses are true, and that then all our cells have multiple mitochondria in it, yes, it comes to the conclusion that we are part of a symbiotic relationship between these bacteria that date back 3.5 billion years ago, and that our constitution is primarily from that relationship, that symbiotic relationship. That's what we are, primarily bacteria in those symbiotic relationships. And of course, concepts like that are concepts that create a lot of questioning and anxiety because it has a lot of implications. But from an evolutionary process and what we're learning with reactive oxygen species, we're learning more that yes, there is science and support to these new theories, which 20 years ago didn't exist. Just to summarize that, at one point, and this they put here in this caricature, they have, you know, the Proterozoic and the pre-Phanerozoic period, all the different periods of time. They have the Eubacteria come together with the Archaebacteria, and then forming that first protocysts, and then how it divides, and they acquire mitochondria as a chloroplast and into plants, fungi, and animals. It's the new theory in evolution, and you can see them here, all these different life forms, and how red algae here, animals and fungi, and plants, how they divide. So the theory that I'm going to show you now is people that have been looking at fossils and looking at what has happened with the progressive increase of oxygen in our planet. And so just think for a moment, Mars doesn't have life, at least what we can see from there, and they're devoid of oxygen. But let's suppose that you started to generate that process of evolution because there was water on Mars and you were able to introduce cyanobacteria. And so what they wanted to look at, and this is the rise of oxygen over the past 205 million years, so they're not going that far back, and the evolution of large placental mammals. His question here was very interesting. Why do we have placental mammals? When did placental mammals occur in all this process that we have of evolution? And so this graph is a graph from that paper, very interesting. So you have the Jurassic, the Cretaceous, and the Tertiary. And then here they have this looking at these different periods and the size of mammals. And then they look at the oxygen concentrations. And they calculate that from carbon oxidation and oxidation of metals, of iron. And they're able to look at that pretty precisely at that present time. And they show that how progressive it started. You know, you had a fraction of inspired oxygen of 10% here, then it went to 15, and only when it came to 17% is when you had placental mammals appearing. Before there, no placental animals. The only one that existed before this was prior to this, and they predominated on Earth, were marsupials. But you know that the marsupial, the fetus has to leave the vagina and it has to be out exposed to oxidative stress, and they go inside the pouch and then stay there. Somehow that was the predominant way of surviving, but when the exposure of oxygen went to 17%, yes, went to 70%, it seemed to be not the most efficient way to survive. And all the multiplication that burst in the formation of different mammals occurred after 17% oxygen. And with respect to the size, now they know that in this planet, at least in this planet, at one point in time, the concentration of oxygen went up to 28%. And that increase in the 28% produced was during the time of the gigantic mammals that we had, the mammoth and all these giant mammals. And then for some reasons, that many explanations for that, the level went down again, and it's been stable around that 21%. But we know that we can affect that concentration of oxygen. And so from an evolutionary perspective, there's always the possibility of change. And currently, the ozone layer, which protects us from those ultraviolet lights at this present time, is being affected also by the way we do things on our planet and how that can affect also the increase in these ultraviolet lights. So this is the typical concentration that we have now, that we all know about, of oxygen, and how we have all this nitrogen, 21% oxygen, and then argon and other gases. And now there have been studies looking at what happens to the fetus and the exposure of the fetus to oxygen. So we can manipulate that by manipulating what we do with the mother. And so in this animal model, what they did basically was expose the mother of that fetus mice to high concentrations of oxygen, normoxia, and low concentrations of oxygen. And what they're able to produce, and they can reproduce, for example, with hypoxia, they've been able to reproduce cleft palate defects. And with using medications that uncouple oxygen, like phenytoin, they can also produce caudal regression in many of these animals. So with hypoxia, you could produce damage and you can kill the fetus. But with hyperoxia, nobody really had studied that. And here you have one with hyperoxia, normoxia, and hypoxia, and they use this medication that's a derivative of imidazole that stains the tissues depending on the level of hypoxia. So this one with hypoxia, you can see all the tissues capturing that stain because all these tissues are very, very hypoxic. So, and this is normoxia, where you do have stain of certain areas like the heart, the liver, the vertebra, and then you can see that hypoxia in the fetus is necessary, and we understand that, for the growth of the fetus. When you expose the female to large concentrations of oxygen, what they've been able to show is that you detain the growth and the development of the fetus. And so somehow in the creation of placental mammals, it was understood by nature that we needed to be able to evolve to what we are as mammals. We needed to be exposed initially to a relative state of what we call hypoxia. And it's in that relative state of hypoxia where you have all the growth hormones, all these growth factors being upregulated. Once you expose to hyperoxia, then all those growth factors are blunted. And so there is no development of that fetus. So we are very, very dependent, absolutely dependent on those concentrations of oxygen. You just heard what we were talking about, oxidative phosphorylation for the brain. So the oxygen and the formation of oxygen species are fundamental to that process, but we need to be able to have mechanisms that counteract the negative effects. But hypoxia, from this perspective, is something that is necessary, but it has a limit. Below that limit, then you produce damage to the fetus. And so here again, oxygen-free radicals are a good example of how useful they could be. This is a cell that, a macrophage that you can see is extending its arms to engulf two bacteria. And then it brings those bacteria into their cytoplasma, and then it releases the active oxygen species, hydrogen peroxide, to kill the bacteria. So it's innate in us as eukaryotes to use oxygen radicals to protect ourselves from the bacteria that can produce damage to our cells. And so the whole process that we think of that produces chronic lung disease, that can produce brain damage, that can produce hypoxic ischemic encephalopathy, it's something that we have embedded to be able to even protect ourselves. We can't move away from this. And different animals have different ways of managing the way they incorporate oxygen and how they eliminate CO2. And very innovative forms, and I think the diversity, and one of the things that these researchers are looking at, because they believe that meiosis occurred at the same time that there was fusion between these two types of prokaryotes to form the first protocyst. Meiosis created the possibility for immeasurable amount of diversity, of genetic diversity. And so we have different ways of being able to use our environment to get the oxygen that we need and to be able to get the nutrition. And of course, going back to the fish, their system to obtaining oxygen from the water is 10 times more efficient than what we can do with our lungs. But down that process, there is this fish that they're doing a lot of research in. It's lungfish, because this fish has both gills and has lungs. So genetically, something happened in this fish that facilitated that process. And it was believed that during times of lower water or higher water and the stress of the environment, some genetic mutation occurred to this animal that permitted them then to start developing what we understand today as our lungs. And if you imagine from the foregut, it's the commonality that your respiratory tract has to develop simultaneously with your respiratory tract, because it was the need for eukaryotes to be able to take in oxygen and to take in carbohydrates from the environment that created that simultaneous development in the foregut. They don't occur differently because there were needs that had to be met as priorities at a time where there was a very stressful environment. And so we get to our amphibians, and then we get to all the different types of antioxidants. We also see that in the cytoplasm of many of these cells, there are also antioxidants that help protect the mitochondria and the organelles from oxidative stress. But that, of course, is all what we consider the hypothesis. It's part of that evolutionary process. So hyperoxia, hypoxic states, oxygen radicals, antioxidants. And so I'm going to jump, there's a balance between the benefits and the negatives, as we've talked about, the double-edged sword for everything. It's embedded in who we are. But what I'd like to go down to, I think everybody has always thought, why do we live in such a hypoxic environment in utero, and then we come out and we say, oh, 21% oxygen. That's hyperoxia for that fetus. That fetus is not accustomed to that, but that fetus is being prepared to that 21% hyperoxia. If we were thinking that 2.5 billion years ago, the highest concentration of oxygen was 3%, 21% took almost 4.5 billion years for our planet to get to that. And then we are exposing premature babies to 100% oxygen immediately. It doesn't seem rational. So that comment of the doctor that said, oh, they're not giving oxygen to that baby, it's unethical. We expose our babies to hyperoxia when they're born on 21%. And this is basically shown, if you look at the venous part, you see the PaO2, 28, and you look at the saturations in the venous, 70%. Well, very different from what it happens when it comes out. There's a big stress and a big challenge for the baby. So at one point in time, we might make that worse by exposing that premature infant to 100% or 80% oxygen. And we're now just starting to learn why that can have such an effect. And if you understand the concepts of reactive oxygen species and how they can denaturalize your DNA, then you can start thinking of how you can affect long-term with resuscitation of 100% of a premature baby, how down the road that can translate into different diseases if you've made changes in the DNA. And this is a little bit of what I was showing you. I talked to you about how you can put causal regression, and this is the animal models I mentioned to you with phenytoin and here, of course, normoxia versus hyperoxia. And normoxia, 21%. But if you have hypoventilation and you're exposed to 100%, your PaO2 goes up to 600. With diffusion diseases, you can get it up to 600. VQ mismatch is similar. The only way you can't do that, even if you go to 100%, is when you have shunts. But in general, we did a lot of that. For example, in our babies with pulmonary hypertension, for many years, we said, okay, if you put them on nitric oxide and that baby has 180, 240, that's good. Wait there. And you can, to be sure that the pulmonary hypertension goes away. And then you would have a baby for 3-4 days with PaO2s above 100. Was that good for the baby? Now, even if it was a term baby, we think that it was not a good thing to do. But at that time, we felt that having those PaO2s would protect the baby from going back into pulmonary hypertension. And so we've had to change our protocols in our nurseries to help people start bringing down the FiO2. And when we have to be next to the patient, we tell them, okay, you had 150, you went down by 5% on your FiO2, your PaO2 dropped to 78, so the automatic just go back on the nitric oxide, go back on the oxygen. And we said, no, don't do that. Come back in 20 minutes and get another blood gas. And they would come back 20 minutes without doing anything, and the PaO2 was from 78 back up to 140. And so they were able to wean a little bit more. They would come back 20 minutes later, get a blood gas. They suddenly wean these babies much quicker than what we used to do before, where we left them in hyperoxic states for a week, trying to wean them off their oxygen and their nitric oxide. So it does affect the way we practice medicine. And in our institution now, we resuscitate babies with 40%, newborn babies, premature babies with 40% oxygen. Is that right? We still don't know if that's the right amount of oxygen. And so there's still a discussion back and forth, and I think that the research, little by little, is bringing us down lower and lower. But it's hard to change and to move from being used to doing 100% to doing less than that. These are our saturation guidelines, and I'm going to end with this. And how much support do I have for this that I'm presenting to you? Where is the evidence to this? And so this is a new change that we did in our unit just a couple of months ago. And we did this change because we went over all the data again, reviewed the papers, the stop-drop study, all these studies, and the support trial and all. And we looked at that outcome that they had with the support trial where there was increased mortality. And so we couldn't maintain those protocols that we were having. In some hospitals, they started to go up higher on the saturation, the settings, with the idea of trying to decrease ROP at the expense of increasing chronic lung disease. Again, the double-edged sword, what is it that you want? And sometimes you go to a position where you're going to have a little bit of both, but you're not going to ever be in the good, beautiful world where you take away both outcomes. You have to pay the price for anything that you do. And so this is what we're doing. Birth weight less than 1,500 grams and less than 33 weeks post-menstrual age. Oxygen target saturation range, 90 to 95. And a long limit setting, 89 to 65%. So you can try to compare that to what you do in your units. Yes. And a long limit, I'm sorry, yeah. That's the upper limit, it's 95. That would be bad, that would be really bad. 89 to 95%. Birth weight greater than 1,500 grams or greater than 32 weeks, post-menstrual age, oxygen target saturation 22 to 98, and the alarm setting goes up from 89 to 91 and from 95 to 99. And it was based, after we reviewed the best available evidence, and saying, well, of the bad things, I think the lesser is to, of course, have more ROP maybe, but we want to have less mortality. We don't want. And people say, well, we don't see it in our unit. And it's not a good response because you need to do a tremendous amount of a large multi-center trial to be able to see these effects. Individually, you won't be able to measure them because you don't have all large enough denominator. So it's not just what you do in your unit and say, well, we don't do that because we don't see that problem in our unit. You will never be able to see that individually. You're just seeing a little part of the denominator, and that's where I bring that concept in. So I don't know how different is this from what you do in your units. So higher up both. So what is this for this? 87 to 93. To 93. So you use lower.
No, yeah. 88 to 92.
88
yeah.
to 92. Yeah. And where do you set your alarms?
85.
And your upper alarm?
I think 96, 95.
96, 95. And for this group of babies, do you do a difference between less than 1,500 grams and greater than 1,500 grams? 90 to 95. So we have higher on this group.
Yeah, you have a little higher.
And this is the alarm. So what we've learned for this, can I tell you that this is the best? I can't tell you that. I can tell you that this is the best. But this is what we're using. We're starting to separate greater than 1,500 grams than less than 1,500 grams. Ideally, it would be better gestational age, and if you have better data on gestational age, because it's all about maturation. And we know that babies less than 1,000 grams aren't the same as babies less than 1,500 grams. We know that by all that we've been studying on development, brain development, everything. And so one of the problems that we have, and we'll talk about that, is what has been moving all this in our nurseries is that we would like to have one way of doing things for all the babies. And that just simply does not work because every individual baby is a different patient, has different problems. And so you have to tailor your management to your babies. And so these are guidelines, but those guidelines can be modified depending on what the problem of your baby is. And so it does take a lot of judgment to modify these protocols because once we put them, they become like policies and they're very strict. And so if you want to change it for a baby, it becomes a whole problem for our nursing staff. We have to tell them that we need to be able to have some flexibility because in some cases it might not be the best. But we're using these guidelines based on that support. We made that change because of this. What we have learned is, and that study has been very important, is that just very small changes in something like target saturations can increase mortality. And that's amazing. I mean, that's how sensitive we are to oxygen. And so everything that I've talked to you about before, if put into the context of what you're doing in your nursery with oxygen, sort of gives you that concept of how careful you have to be and how important it is to individualize those treatments with oxygen. And that's the message that I wanted to bring to you today.
We can use higher level after a few weweeks, not immediately after birth. I mean, after 2 or 3 weeks, you might give more level of oxygen saturation, but they say that the chance for the drop is less. In the baby is 3 weeks old, so you can give higher oxygen. More than
At what, so you do it by gestation, like when they reach certain gestation?
First day we have less. So 2 weeks or 3 weeks, yeah, you might have more than 9.
Sandra, what do you do in unit? If you can speak.
88 to 93% is what we use.
88 to 93%.
And then we set the alarms for 85 to 95.
85 to 95. And what do you do in your unit?
We were not very impressed about the support study, as you were. And we keep still now, in the majority of the cases, 88 to 93.
And they were saying that they were planning to do symptoms instead of whatever.
But Marek, do you know, it's very interesting. I think the evidence was very solid that lower saturation limits were better in terms of EPD and ROP. The problem is support study, actually.
Yes.
And the question is, what is the reason, what was the cause of death of these babies? That would be very interesting.
Yes, and we looked at that study. I think all of us went to find out why these babies died, and we couldn't find a good explanation for why there was a higher mortality in those babies. And of course, if you look at what we're talking about, all that we've talked about, oxygen radicals, we do believe that oxygen radicals are in that. How much they're influencing mortality and how they're producing that mortality, we don't know if this was to be reproduced. So you'd need another randomized control trial like that, showing that, you know, those SAT limits, when you use the lower SAT limit, you continue to have high mortality. Where you find a cause and effect relationship, I think that's very important, but we don't know that. There is a very interesting study. We were talking today about the study, that randomized trial looking at cooling versus no cooling, and how they were able to find increased survival with less moderate to severe brain damage. One of the things that they found when looking at the data more in detail was that in the control group, there was a large percentage of babies that were being overheated in the control group. And we know now that overheating, hypoxia, and exposure to oxygen produce much more oxidative stress. And so we think that the control group is looking worse, not so much because they weren't exposed to the cooling, but because they were being put under warmers and having overheating. And that's been shown in animal models also to decrease and make the outcome worse because of more severe oxidative stress. With this, we don't know. We need to find out. But when you look at randomized control trials, I think the good thing to go back and look at the support trial is to see, you know, in that large number of babies, because it was a large number of babies, they did do those randomizations as well to these targets. How well they kept them, and that's a different question, and we'd have to go to the investigators. You know, how easy was it to maintain these alarms and these set levels in these patients? It's not easy. We all know that it's not.
The difference was actually 3 points on the main saturation, if I remember correctly. And the other question I would have is, if we assume that low saturation is dangerous, why to change both limits, the higher limit and the lower limit? But in this case, I would say, okay, let's put the lower limit a little bit higher, but keep the higher limit still by 93, because after 93, you cannot be sure how much pressure of oxygen you have. It could be 100 or 150.
And those are all good questions, but then you have to do the trial with those. And so, for example, in our guidelines, if oxygen saturations exceed the high limit for either group, wean O2 concentration by 2 to 5% every 2 minutes. Stop weaning when oxygen saturation reaches 90% for babies less than 1,500 grams and 94% for babies greater than 1,500 grams. Remain at the bedside until the desired saturation range is reached. Now, This is good at sea level. But if you go do this in Colombia, where we're at 2,600 meters above sea level, you know, a protocol like this might increase, you know, chronic lung disease. There, of course, because of the altitude, these saturations are lower. We had, in our study of CPAP, a higher incidence of chronic lung disease according to the definition of 36 weeks post-conceptional age. But it was in Bogota, when we compared it to other cities, it was only like 13%. In Bogota, it was 50% because of the altitude. And so when you start manipulating saturations at that higher level, the range for producing damage is much more narrow. More likely, these changes are tolerated better at sea level. But if we reproduce something like that in Bogota or in Colorado, maybe at high altitude, then you might be able to see even more mortality. And that's a hypothesis. But again, you're going to hurt on both sides. Where is it that we find the balance? And so we don't know, and I don't have a response to that, but we are becoming more sensitive. We're working with our nurses, so they, as soon as the baby has an apnea, the first thing that they always used to do, you know, they were 25% and suddenly 80%, and said, my baby's doing very badly, what's wrong, 85%? No, it's that the baby had a little bit of an apnea and desaturation, and so the babies are being swung back and forth between these changes in FiO2s, and we're trying to educate our nurses to understand that that's not the way you should respond, and so we're working on that.
One of the things that we've been talking about a lot in our country is changing the SAT limits, not so much for birth weight or gestational age, but for postnatal age, so that when the babies are no longer at risk for ROP, if you have BPD, to put them in a higher oxygen range. Thinking about the risk for core pulmonale. Again, no evidence in particular, but yeah, it makes some sense.
And it makes some sense, and so that's why you would like to be able to do that randomized control trial.
If I go back to my CPAP pill and put mine to around 5, you will kick me out. I'm sure about that. It's always shocking.
And so,
88 is good.
So if I told them, okay, is that an opinion or is that based on evidence? And then if you were able to invite them to a dinner, you invite them to a dinner, yeah, and you sit them down together,
Dinner with him.
yes, and you look at the evidence.
No, because there's always shouting. When he sees saturation 92, what are you doing? Killing the baby.
And so that's where we have to ask.
90, 88, 99, that's all.
So,
When the baby is more than 3 weeks, 4 weeks, 8 years, will not be shown.
And so if you ask me, you know, would I be so passionate about that? I would say absolutely not. I'm not going to be passionate about something that's so filled with uncertainty. And so what we have right now, the best randomized control trial, even though many people might disagree with the CPAP way they designed it or everything, but that randomized control with 1,300 babies and how they did this and how they were able to find mortality, you have to pay attention to that information. What's better than that at this point in time? Show me an objective study that guides you more than that study at this point in time, and we don't. We have a lot of questions that need to be. So if somebody asks you what's the best available evidence to decide on what target range you should use in a nursery, I probably would, and everybody would be saying, well, we have a Class A randomized control trial with 1,300 babies where they randomized them to 2 SAT targets and they showed increased mortality with decrease in ROP. And it seemed to be a very well-designed randomized control trial. And so it's hard for me to contradict that information and then not take that information and try to rationalize that. Is that the best? There are more questions that need to be answered. But your best available evidence at this time is this. And I cannot be passionate about this because here's where you start to think I still have equipoise because the information is not good. This is trending towards telling me that maybe lower SAT ranges at sea level are not a good idea. But we need more of those studies to be able to support that. If that is consistent with other trials, repeating that trial, and then our evidence becomes stronger for everybody to change because you can't pay a cost of mortality with an intervention that you're manipulating in the nursery, if that's the outcome. If the outcome is ROP, higher ROP, you're going to have to accept, yes, maybe higher or ROP. It's better than mortality. But you won't get it both ways. And so what I would tell anybody who's passionate about this is that's dangerous to be passionate about something that's so uncertain. I wouldn't recommend that.
What were your limits before you changed it?
Hmm?
You said you just changed your limits. Where were they before you changed it?
We were doing the same thing that you were doing. Yes, and so we were using the lower sat, yes, and those babies that we were concerned more about ROP, because we were focused more on the ROP issue.
Why do they need more oxygen? I mean, you're less worried about ROP for the big babies, but why do they need more oxygen?
Well, that's what the
I mean, they don't.
right. But again, do you see where we came from? The support trial, because the support trial showed that there was increased mortality when you use the lower saturation range. So with that randomized control trial, we felt that we couldn't continue to expose the lower group to higher oxygen concentrations.
The big babies, I don't understand. Why do they need more? I mean, it might be they have less probability to have a ROP, but if we know that oxygen is toxic, it can be toxic with high exposure, maybe they can be fine with 95.
And you're perfectly right in saying that. But that's how uncertain, this is where we are. We're in the terrain of uncertainty. So I'm not passionate about supporting this at all. You're not going to see me killing myself to support this at all. There are good arguments in favor and against it. There are very good arguments, and arguments that look at different gestational ages. At Vanderbilt, where I was before, we took the gestational age, and we were looking at it's very different when you're less than 32 weeks, greater than 32 weeks. We're going to change what we do, but we were not basing it on any really good, strong evidence. And so, we have one study, not the best study, but that's the best available evidence, and we have an outcome that's concerning, which is mortality. And so that's how we start to use that information. We want to have more information. But so now you have something to do a nice study and come up with the answers. Here in your unit here, what do you do?
Approximately the same rate for the lower at 88 to 93 approximately. And the 90 to 96 for the other. But I think that the problem is that it's not the target, but how you manage the limit. Because that's
Yes.
when the story.
And that's the hardest part, where we have the education, and with our nurses we have little cards that they have to carry around where they have this, because it's very difficult. You may want to have that occur consistently, but it's very, very difficult to do. You have to be present all the time. Now there's a new technology that's coming out where it has an alarm system that tells you very quickly what your trends are with respect to how much you're hitting the higher targets, how frequently you're doing it, so that you can see if you're really educating the people appropriately, or you have to go back and re-educate and supervise more what's happening. But it's very time consuming. Okay, thank you very much.